So much for such a comprehensive introduction. I feel like I can then just jump right in. That was perfect. So, today I'm going to talk about the aquatic subsurface. I was looking at the different lectures that you had, and a lot of them were marine focused, some freshwater, and then the cryosphere presented by Christine, who we just heard I did my PhD with. But beneath our feet, there is a very large aquatic habitat. So, just a little bit about my research interests. I have really broad environments that I study, but at the core of it, I'm a microbial ecologist and I have applied and fundamental research interests. So, today's talk is going to be more on the applied aspect of my research because I think it's important for people at all different stages of their career to understand how this fundamental environmental research that we're doing actually has an applied aspect that can help us solve real world problems. So today's talk is going to be more applied than maybe observational investigative. And so the types of environments that I study range from glaciers that are in Montana. So these alpine glaciers, high alpine glaciers, glaciers in Antarctica, in Alaska, to groundwater aquifers. I study rivers, lakes, and streams. And I also follow this up with detailed investigations in the lab with different reactor systems, as well as looking at how organisms grow in attached community assemblages. So let's start off with the importance of the terrestrial subsurface. So what is the subsurface? The subsurface is characterized as the environment that starts directly below the surface of the earth. So this means 1 centimeter down, that's considered the subsurface. But typically when people think of aquatic habitats, we're thinking of marine systems, lakes and rivers. But actually the subsurface is a large reservoir of natural energy resources and of water. So that we know that water covers 70% of the Earth's surface, and only of this 70%, only 1% is available for human use. And 95% of the water that we would use for human use is freshwater and available, and it's contained as groundwater. And so this groundwater, these reserves are becoming more and more threatened as we increase the world's population, the need for agriculture, the need for increased food production. And so what's going to happen as we start to deplete and contaminate these resources? One example that I'll come back to is the loss of the Ogallala aquifer in the United States. So this is an image from a review paper that is the slice of the subsurface, and you can see starting at the top, oops, my mouse is very sensitive, sorry about that, ranging all the way down into the deep subsurface. So if we start at the top, we have these shallow kind of porous matrices, and as we go down, there's an increase in temperature and pressure. But what's in common across all of these different layers is that there's a lot of porosity. And porosity is important because this is where the microbes live. This is the space in between particles or rock pieces, which is depicted over here, where microbes can actually inhabit. It's important to know that about 1/3 of the biomass on Earth actually resides in the subsurface. Microbes have been shown to exist in the subsurface across a wide range of rock and habitat types, but we know from few studies, there are some, that biomass, the typical trend is that biomass is going to decrease from the top to the bottom. But we do see what we call microbial hotspots. So while we're seeing an overall trend of biomass decreasing with depth, we'll see these hotspots where we'll see a proliferation of microbes. We're not always sure what's driving these hotspots and what this means ecologically. So today I'm going to be talking about two different subsurface environments. One is shallow and one is deep. So the shallow system I'm going to be focusing on is a groundwater aquifer. And then the deeper system is going to be on microbially produced methane to be used as a natural gas research. So what distinguishes between the shallow and the deep? Some would say depth, right? So shallow is closer to the surface, deep is closer to the bottom, but actually the technical characterization is based off hydraulic connectivity. So you can see that up here, we have this kind of vertical flow for groundwater. So this would be the shallow system. And then we see the bedrock, and there's actually a whole different hydraulic regime down here as we're deeper. So I said this on the previous slide, but we're going to talk about a terrestrial groundwater aquifer, and then we're going to talk about coal bed methane. But across these two environments, and pretty much all the environments that I study, what I'm interested in is how to identify the active microorganisms. So I know you guys have had a lot of talks on this, but instead of just going out to the environment and collecting a bag of genes and seeing who's there, how do we actually start to discern these function relationships and what microbes are responsible for specific biogeochemical cycles of interest? So back to this Ogallala aquifer. So the Ogallala Aquifer is located in the central United States. It's what's called the breadbasket of the US. This is where 1/6 of the world's grain production comes from. So of the world, it's a really big producer. And what we're doing on a continual basis is we're draining this aquifer. And this aquifer isn't just necessary for human consumption, it impacts ecosystem function as well. And we're part of the ecosystem. Fish are part of the ecosystem. So what happens when we drain these aquifers? Well, what you can see here is that the natural bedrock where this aquifer is overlaid is actually really high in nitrate and really high in uranium. So as the groundwater starts to decrease, as we're producing more food, we start to get a release of heavy metals such as uranium, which is very toxic to humans and other animals, as well as nitrate. And nitrate is really bad for humans because it will scavenge oxygen in your blood, and it causes what's called blue baby syndrome. So you're actually suffocating yourself from the inside. And what they're showing is as we're depleting this aquifer, we're actually seeing an increase in uranium concentrations in our drinking water and in nitrate. And they project that in 50 years, 70% of this aquifer is going to be depleted. So this is a big deal. What do we do with the loss of this freshwater resource? And then what happens, can we remediate this water that has such high uranium and nitrate concentrations? So where we go to study these questions of how can microbes help us to remediate nitrate and uranium concentrations is actually at the Field Research Center in Tennessee. The FRC, or Oak Ridge, was a site, a processing plant that was in production during the Manhattan Project. And for those of you who don't know what the Manhattan Project is, this was part of World War II when they were starting to develop nuclear weapons. So that this site, they were generating plutonium and uranium for these nuclear weapons, and they were dumping the waste into these ponds right here. So these are called the S3 ponds, and they were waste reservoirs for all of this extra material. And so then what happens is these materials leach into the groundwater. And today we have incredibly high concentrations of heavy metals, uranium, and nitrate. So this is kind of a great natural laboratory, well, man-made laboratory to study these questions of what could happen as we start to deplete these groundwater resources. And the questions that we're asking were from the fundamental side, from a project funded by the Department of Energy, is what microbes are there, which microbes are active, and how do these different microbes respond or change to an environmental perturbation. So if you have an ecosystem that's functioning in its natural state, circumneutral pH, low concentrations of heavy metals, what happens then if they get this influx of heavy metals to the community? So we're really interested in dissecting these dynamics. So this is another view of the FRC, or the Field Research Center at Oak Ridge. And what I want to highlight here is that terrestrial aquatic ecosystems are very complex, and we often defer to them in 4 dimensions. So instead of going to a lake where you're taking an aliquot of water and maybe you just have to deal with where you are in the water column, the stratification, we have things that are coming in from all different angles. We have geology, hydrology, different flow paths that we have to take into consideration. So really this field is a little bit farther behind just because of these logistical and environmental constraints that we have. So some of the questions that I've been involved in answering is who constrain or what constrains, so what environmental parameters constrains the organisms that are there, which organisms are active, because this is a flow through system. So it's constantly changing and it changes on a very fast scale. We'll see a complete refresh or recharge in under 4 hours. So, are those organisms just passers in the groundwater, or are they actually contributing to the biogeochemical cycles that are there? Or, is the groundwater even important to study? What processes are active and why are these specific processes active? Are there any interactions that constrain the microbes that stay versus leave? And then at what scale? Scale is a huge question for us. Are these communities constrained? So I told you about the really high temporal variation, but we vary across space as well. There's very high diversity, but communities that are even a meter apart look completely different from one another, and they have the same exact source water. And so we're trying to understand what's changing these community compositions. And our overall goal for this is to get at something that's being referred to in the field now as mechanistic ecology. And mechanistic ecology is, how do we take all of these observations that we're collecting as microbial ecologists? So we measure pH, we measure who's there, we'll measure rates of activity, and how do we turn this into a predictive mechanism? So if I were to get a rainfall event, or if I were to see a change in pH, can we then, from all the information we've collected, predict what the community response is going to be? Now, this is a really, really lofty goal. And if we could do this, it would be amazing. And maybe none of us would have jobs anymore. But this is the goal that we're trying to do, is combine all of this data into essentially a model, which is what I'll end this section with. So just a little bit of information on the different zones because they're important. The subsurface is stratified just like a lake or an ocean, where we start at the top, and this is called the vadose zone. And the aquatic aspect of this zone would come from precipitation infiltrating down when it rains, or evapotranspiration. And then below the vadose zone, we have what's called the capillary rise or the capillary fringe, and this is the transition zone. So as the groundwater table height changes, this zone will be intermittently wetted and it changes in height. And then we have the saturated zone, and this is where the groundwater is, and this always remains wet. And so in a paper that I was on, what we determined was that we think that communities that are in this saturated zone are the most stable. They're not changing over time because they're not going to be experiencing the fluctuations in environmental pressure. But when we look at this capillary rise, where we have that intermittent wetting and we're seeing a change in the different environmental parameters, this is where we think that we have the least amount of stability in the community that's present. Interestingly, what I'm going to show you in the next slide is that, or in a couple of slides, is that this is also where all, I would say 90% of the activity is. So what is it about this zone, this intermittent wetting zone, that is driving biogeochemical processing? This is where we see total activity, high nitrate respiration, high sulfate respiration. And so we don't understand why the activity is located at that point. So to investigate this idea of environmental pressures, I was involved in an experiment where we drilled two new cores, one that was in a circumneutral pH, so our natural background uncontaminated site. So this GW277, we drilled this core all the way down to the bedrock, and then we were collecting corresponding groundwater so we could understand the processes that were happening in the groundwater as compared to what was happening in the attached sediment fraction. We then compared this to a highly contaminated well, which is EB106. So this had low pH. The pH changed from the top to the bottom from 5 to 3. And then you hit this 3 very acidic conditions once you hit that toxic waste plume. So the groundwater in this site was actually the toxic waste plume. It has extremely high nitrate contamination, from ranging from 10 to 100,000 parts per million. So this is higher than in any natural system you would see, but yet we still, from our results, we're seeing evidence of life here. And it's important to note that the geology, so we selected these two sites because geology and hydraulically, they were the same. They just had different geochemical conditions. So I think people are a little bit more familiar, and I was before I started the subsurface work, with how to sample in aquatic systems, ocean, lakes. And you can collect a lot of samples, but there's a lot of logistics to collecting a core because you have to then divvy it up and over what increments. But I just wanted to show you that for this experiment, we collected a lot of samples. It was over like 5,000 individual samples in total for these two cores, where we sectioned the cores into 9-inch segments. And then all of these different analyses that you see here, we had to take from each core segment. And that was just, we're collecting all of this data because we don't have an idea of what information we need to be able to get to our goal, which is that mechanistic ecology question. How do we understand what's going to happen to a community when they see a pH shift? So we went into this kind of blind and we were just collecting everything that we knew how to measure. And I'm admitting that because that's what that's what people do. So when you read a paper, usually they go out to the environment, they sample it, we take these observational measurements, and then from that and statistical analyses, we try and discern what's happening or what are potential drivers. So we're in the same boat here. We're just collecting everything we could to try and describe this system. So one of the main parts that I was, well, I was involved in the whole project, but coming back to the active fraction. So I think it's well accepted, and this group knows this. I'm preaching to the choir here that we cannot cultivate a majority of organisms that are in the environment, but how do we study them in a representative natural state if we can't culture them? So this is of enhanced importance for the subsurface because they're under pressure, there's different temperatures, and it's challenging to replicate these conditions in the lab. Um, so what I tried to do for you is, I think that this is a nice resource when you're thinking about your own research or projects, or when you're even reading a paper, is how do people currently study the active fraction? And I think that from my perspective, this can be divided into two different categories. It can be divided into tracer-based approaches and non-tracer-based approaches. So for this tracer, this would be where you have to take the environmental sample ex situ, bring it back into the lab, and you're adding some kind of tracer. And this ranges from stable isotopes to radioisotopes to different fluorescently labeled substrates, or something that's new that I'm really excited about, I'm not presenting on today, is this substrate analog probing. And then there's not, so this, there's going to be some alteration to the community potentially as you incubate it. It's not maybe as representative of the natural state, but there are a lot of positives that you get from this. And I'll talk about the tracer-based approaches in the next slide. Then there's the non-tracer-based approaches. So this is where you're taking material and you're analyzing it directly from the environment. So this usually consists of some type of genomic analysis or other biomolecules, and methods are metatranscriptomics, metaproteomics, metabolomics, single cell genomics. You can have different intercalating dyes. There's amplification-based techniques, looking at 16S mRNA, protein coding genes such as Rubisco, or other functional genes, AMOA, involved in ammonification, or sulfate reduction genes. So you really have to decide when you're picking these methods, what are your questions that you want to answer? And then are the limitations and the biases, how do you overcome that to be able to feel confident in the conclusions that you're making? So I do both. I use both tracer and non-tracer methods, and oftentimes compare the two, and you get, spoiler alert, you get very different answers, and it's very challenging to compare them. But you learn something from each technique. So for the tracer-based approaches, they can be broken down a little bit further. Some can be quantitative, so you can get rates of activity. I've done a lot of work with radioisotopes. Radioisotopes are the most sensitive because of our detection capabilities, but this is a bulk technique. We don't know which organisms are necessarily responsible for the activity, but we can get a rate. Then there's stable isotopes. This can also be bulk. There's extracellular enzyme assays. And then on the single cell level, as I know you guys have heard about, there's nanosims, Raman microspectroscopy, and then MARFISH, which can be semi-quantitative. And I would argue that Raman microspectroscopy combined with stable isotopes is also semi-quantitative. And then there's the phylogenetic identification. So these aren't quantitative techniques down here, but you start to get more insight on which organisms are responsible for that function. So there's DNA with qPCR, stable isotope probing. There's SAP, so this is the substrate analog probing. So you'll feed it a synthetic substrate, And this includes BONCAT, or bioorthogonal non-canonical amino acid tagging. It's a mouthful, but I'm going to go through what that technique is because that's what I've been focusing on for the last about 4 years. So this is community level where you then sequence, for example, the 16S, or more recently, we've been combining some of these techniques with helping in metagenomic sequencing. And then you can get to the single cell, and single cell studies are amazing. They offer unprecedented resolution. But again, I want to say that it really depends on your question. If you have a really focused question, I think that that's when these tools are really appropriate. But if you're going to an environment for the first time and maybe you don't know who's there, maybe you need to bump up a little initially and start at the community level. So here's some results from that coring study that I was telling you about. Um, And what you can see here is this is some of the different activity assays that we had. So this is from the uncontaminated core. The yellow color is our vadose zone. So that's that mostly dry zone. Then we're going into the capillary fringe, and where this upside down triangle is, this is the height of the water table. And then we're into the saturated zone. And so what we can see here for general activity is we're seeing, this profile over depth, much like we would look at a profile from the marine environment, where we're seeing some spikes up near the top, but then it kind of stabilizes. Not much is happening here. And then we see this spike happen right at the transition of the water table. We also see that spike correspond with our sulfate reduction activity. And it's not a perfect overlap, but we see nitrate reduction leading to nitrous oxide off-gassing also happening at this water table. So this was the first time that people have looked at this fully comprehensive stratified core and trying to understand where the activity is, where should we target when we're doing these bioremediation efforts. And we've really focused in now for future studies based off this on this water table at the capillary fringe. And so we used techniques to look at the bulk rates of activity. This had to be an ex situ, where we obviously took the core out. We incubated with tritiated leucine for very short times. That's one of the benefits of radioisotopes, is that it's very sensitive, so you don't need a long time. And sorry, I have to keep moving the video so I can see where my slide is. So at the top, this is our pristine core. We see this spike in activity at the water table. And then at the bottom, this is the contaminated core. And really not a lot is happening as we go down. We're seeing a lot of dead necromass down here. So the organisms are very, very lowly active as we continue down with depth. But we do see this spike right here. And interestingly, this spike in biomass it corresponds to the presence of clays. So there's something about clay that's enabling organisms to maybe survive or proliferate one of these hotspots that we were picking up on. So I talked about two compartments in the subsurface. There's the sediment attached fraction, and then there's the groundwater. And so what people typically do is they'll go to the subsurface and they pump the groundwater because it's a lot easier than drilling a whole core. And then they'll try and infer what's happening in these systems from the pumped groundwater. And it's been kind of a long-standing question is, what's more important in these systems? Is the groundwater representative of the sediment? Where is the bulk of the activity coming from? So what we directly did was we compared rates of activity from the sediment to the groundwater here. So here we can see a couple of activity rates as we go down with depth for the sediment. And then in the graph on the right, I'm highlighting what the groundwater activity would be. And this is challenging comparison to make because sediment is on a per gram basis and groundwater is on a per mil basis. So how do you compare across these two different volumes? Well, we converted it to a per cell basis. So we know that there's different numbers of cells in these two environments, but on a per cell basis, we know that cells in the groundwater are actually more active than what's happening in the sediment. I think that this is a really interesting finding. And my conclusion right now from lab-based studies is organisms on the sediment aren't going to grow at a super fast rate, because if they did, they're going to clog off their nutrient resources. So you can think of it as a pipe. If they were to overgrow, then water can't infiltrate through. So there's actually the thought that organisms that grow attached to particles are preconditioned to grow slower than planktonic cells. And then if we look at the contaminated core in this bottom graph, we see low levels of activity. Surprisingly, it's about 2 orders of magnitude lower than what we see for the uncontaminated site, but we're seeing in the same order of magnitude. The groundwater population doesn't have the same protective mechanisms as attached fractions do, but it is still active, but much less so than in our pristine site, which isn't surprising. But these organisms are active. So we were thinking, well, if they are potentially active, if they were to go somewhere else, would they then colonize a downstream ecosystem? Are these organisms endemic to this highly contaminated site? Um, but what we're seeing is that, we know that they're active across both fractions, but what does this mean on a larger scale? Does it, are groundwater organisms actually important to biogeochemical cycling? And the way to do that is we did kind of a back of the envelope upscaling mathematical equation where we took a cubic meter. So if you were to take a cubic meter section of this groundwater and we calculated the porosity, so how much available space would there be for water? We calculated how much water there would be compared to sediment. And is the activity that we're seeing, even though it's high on a per cell basis, is this actually important on an ecosystem level? And I'm just going to draw your attention to these last columns here. And so if we look at the top in the orange is the pristine environment, and you can see that at certain depths, maybe 13% of the total activity in this environment could be attributed to groundwater, but it's pretty low. And then the percentage of activity for the contaminated environment is incredibly low. So our conclusions based off this is that really where the important place to study, it's from these attached communities in the subsurface, and it's not actually from the groundwater, that the bulk of activity and biomass is associated with sediment. So we then used this is where we compared a couple of techniques where we used PMA, which stands for Propydium monoazide, and BONCAT. And PMA, it gives you a proxy of potential viability. So it's an intercalating stain that you add to your samples. And if the stain's allowed to cross the cell membrane, it will cross-link with the DNA through a UV light treatment, and then that DNA can't be amplified. So if it can penetrate through the cell membrane, we say that those cells are compromised. So they're potentially dead. They're not going to be viable. And then with BONCAT, it's actually a tracer-based method. And what the conclusion is from all of this sequencing is that essentially it was 99% of groundwater organisms are potentially viable. It's not dead necromass. There's not just about a bunch of dead cells floating around, that they are potentially viable. But what we saw was very different results from what was potentially viable and what was actually translationally active. So of the total community, only about 11% was translationally active. So while they're all, alive, potentially alive, a very small amount of that population was actually active under those conditions, which raises some really interesting questions about what conditions would those other organisms then become translationally active under. What else we can do, we have metagenomes from each of those sections. So what I was talking about, these non-tracer based approach, we can look at the stratification of certain genes of interest. So this is all shotgun metagenomics. So we're able to quantify the relative abundance of different genes. And we see the presence of different genes across sites. On the left, this is our background pristine condition. And then on the right, it's the contaminated low pH, high nitrate condition. And we see a higher percent of nitrogen cycling genes associated with the high nitrate core, not surprising. We're now in the process of doing some co-occurrence analyses on these to understand if there are truly any patterns. As you can see, this is kind of difficult. We're not seeing a ton of stratification. The only stratification that really pops out to me is that we see an increase in nitrogen cycling genes once we hit that nitrate contamination plume. So at where the groundwater actually comes in contact with the sediment. So this graph that's spanning across the screen is kind of showing you all of the different observations we made. And it's not for you to have had to read that, but just to show you, get back to this observational question of, what data can we collect, and then what data is actually important. Yes, we can collect a lot of things, and we have spent 3 years trying to understand what the correlation is across these different measurements. We're working with mathematicians, statisticians, And because my desire from this was I didn't want to write an observational paper. There's a lot of those out there, and I really wanted to get at mechanism and understand what was driving these communities from all of these different measurements we took. And hopefully in a couple of months, there'll be a paper coming out, but it's going to be an observational study because it turns out we didn't collect the samples across the necessary or frequency scale or at the invite scale. I mean, we don't understand those interactions well enough to get at this mechanism. So I'll have an observational paper. It's interesting, but I didn't accomplish my goal of how do I get at mechanism. So what I did was I sat down and said, okay, what would I need to do for next time? As any good scientist would do, how can I improve upon these efforts? And working with our team, we came up with this conceptual model. And this is way more math than I am comfortable with on the right. And so in a perspectives paper where this conceptual model is presented, it should be out in the next couple of weeks, we break down all of the different types of measurements that we think need to be taken across different systems. This isn't specific to terrestrial aquatic systems, but just when you're trying to get at mechanism, what different parameters do you need to consider for your measurements to understand what is driving these microbial community assemblages. And so that's kind of where that study right now is that we're going to go back into the field once COVID is over and try and apply our model. We've in theory, we're putting it out there based off we call it our lessons learned from that really large project, but what do we need to collect next time? And that's what this paper is about. And so I think once it comes out, I would encourage you guys to read it, especially, as in your graduate career, it's really geared towards that level and to kind of talk you through experimental design considerations. So hopefully that's out soon. And I just want to, this is a really large project. These are all of our partner institutions. I presented on a small piece of it because I want to get to the coal bed methane work. I could, give longer talks on all the different measurements, but I want to thank our whole group. This is the people that were involved in it. So now transitioning to coal bed methane. It's no surprise that our world population is increasing, and as the population increases, so do our energy demands. And so what this graph is showing is the different types of energy resources that are currently used, spanning from 1830s to 2010. The red line is the CO2 emissions. So what you see is that with increased energy use, we see increased CO2. I think that's well accepted. And then the black line is the same line from the previous graph that is showing the actual energy demands that we need. And it's important to note that coal is it's a dirty energy. I think we know that. And it's called a dirty energy because it produces the most CO2 out of any of these other energy sources that there are. So it's the hardest on our environment, and we want to move away from just relying on coal. So what is coal bed methane? Coal bed methane is actually a gas that is derived from methanogens in the subsurface. It can be derived, I should be corrected. Coal bed methane can be formed thermogenically or biogenically. Biogenic methane is from methanogens, where thermogenic methane is, over time under high temperature and pressure, peat, it gets converted into methane and then renewable. So when people extract it, once you take it out in this biogenic methane is that, from these wells, we can continually regenerate it. So it's sustainable. The way that methane is extracted, coal bed methane is extracted, you can see this picture here. It really doesn't disturb the landscape too much, as compared to strip mining where you go in and you have to take off whole mountain sides. What you do here is you're removing the water from that pore space. And when you remove that water, the methane comes out simultaneously with it. So this can maintain the land use. This is a schematic that represents the sequence of biogenic methane production. So how it starts is we have these organics from coal. So coal is a really recalcitrant carbon source. So we have coal, and in between all of these, there's a lot of cracks in coal. So in between the cracks, this is where the water is. This is the aquatic habitat. This is where the microbes live. And so over time, the coal will break down, or the bugs themselves can actually release different compounds such as exoenzymes to start to chew on that really recalcitrant organic matter. These are the fermentative organisms that will usually do this, bacteria. So it'll break down these large organics into small organics, into the precursors that are needed for methanogenesis. So there's three types of methanogenesis. So one of the precursors that could be generated or is generated is acetate. And then these methanogens use these precursors and they produce methane. There's a lot of factors that influence how much methane is produced. It depends on, how recalcitrant is your coal, how thermally mature it is, and then what organisms are there down that coal for these methanogens. The methanogens are just kind of waiting for their substrates to flow. And so it's often believed that this is the rate limiting step. And so this is where my research is really focused on, is how can we increase this initial breakdown of coal to get more substrates necessary for methanogenesis? Makes sense, right? More substrates, more methane's produced, more natural gas. We can kind of relinquish our need for coal or other dirty energy sources. So this is our field site. It's in eastern Montana. It's called the Powder River Basin. It's really beautiful. It's different from a lot of my field sites. There's not a lot going on. It's the desert, and these are our wells right here for size reference. So there you can see that there really isn't a lot of impact on the land. We just have these small wells. This geologic feature contains 40% of the coal in the US. So you can imagine there's a lot of controversy over wanting to mine it and dig this coal up. So what we're trying to do is say, hold on, we think we can come up with a strategy that's less invasive to produce more methane. But a lot of this coal isn't accessible, so they couldn't mine all of it even if they wanted to. Um, so this is where it's located. I live right over here. And so it's about a 3-hour drive over to the Powder River Basin. And one of the side effects that, or the environmental impacts of coal bed methane, not the way that we want to do it, but how other people, the current strategies is you get all this water that I was talking about. So when you extract the gas, you're also pulling up all of that water. And you can see from 1999, which is before any of these CBM wells were installed, you don't see any of these blue dots. And what the blue dots are these holding ponds. And they're holding ponds for the water that was extracted. And the water that's extracted from these subsurface environments is very saline, so really salty. So it can't be used to feed livestock, and it can't be used for irrigation. So what ends up happening is we just have these holding ponds that are scattered across the landscape. And, it starts over time. If we were to keep drilling new wells and keep pumping out this methane, we would have a lot of these wells, and that is using the available land space. We don't know what to do with that water. So here's our test site. A couple of years ago, we worked with the United States Geological Survey to put in a battery of 9 different wells that span different redox gradients. And the specific redox gradients that I'm talking about is that there was different sulfate concentrations. So sulfate is known to inhibit methane production because sulfate reducing bacteria can outcompete methanogens. So in the presence of sulfate, we don't see methane being produced. And we want to understand why. What is it about those environments? Is there some threshold of sulfate where maybe the methanogens could start to produce methane? And so at the top, these are our non-sulfate containing wells. So this is where we see methane. And then down here is where we don't see methane production, just breaking them up. So what our overall goal is how can we come up with a strategy? I work with a lot of engineers on this project to enhance biogenic methane production. So is this going to be through bioaugmentation? Do we put organisms down well to try and stimulate methane production? Do we do biostimulation where we actually put a substrate down well to get more methane production, or is it just leave it alone and we have to wait for a really long time? So this is kind of what we're trying to investigate is, what substrate should we put down well? But the first step is we have to understand how are the microbes, the resident community that's there, how are they actually breaking down this coal? How is methane being produced? So in a study that's in review right now, we sampled these, oh, I'm sorry, across these different redox gradients for shotgun metagenomics and for Bongcat metagenomics. So with shotgun metagenomics, we got really high quality DNA, so we're able to be quantitative from these results, know how many genes, like what's the highest functional capacity that's there. But what this doesn't tell us is, are these actually being used? Are these active? So what we did was we combined this with a novel approach, Bongcat, and in the next slide, I'm going to talk about how that exactly works. But in short, it targets the translationally active organism. So compare these translationally active metagenomes or shotgun genomics to understand which genes were active, which organisms were active under these different sulfate and redox conditions. And what's really exciting about this is we were able to collect these samples. So this is called a subsurface environmental sampler. These wells are really tiny. They're about 3 inches in diameter. So you have to get something that's really tiny to go down there. We inoculate this with coal from the coal seam. And then we incubate it for a couple of months so it can colonize down well. And then the doors will kind of close. So when it comes up, the sample is under the same pressure and temperature as it was before, and gas headspace. So it's kept anaerobic until we can get back into the lab. And we always keep it under the same pressure because remember, this is the deep subsurface, high pressure environments. So then these tracer approaches, we really feel are more representative of what's happening in situ just based off our how we collect the samples and then how we process them. So the shotgun metagenomic results, there's a lot going on here. As with metagenomics, you could write 15 papers off one metagenome. So you have to decide what your questions are and what you want to ask. But what I want to familiarize you with is we dramatically classified all of this data into MAGs or metagenome assembled genomes so that we could assign which genes go with which organism. We had really high completion. We only looked at the highly complete bins. Some people will look at stuff more in the weeds, but our cutoff criteria is that each metagenome assembled genome needs to be 5% complete so that we feel really confident that belong with the genome. That is one of the biggest problems right now in metagenomics, is that we're making inferences on organisms that are computationally reconstructed, and there's a lot of contamination in those genomes. So we've been working on methods bioinformatically to really decrease that noise or that contamination. So for Bongcat, bioorthogonal non-canonical amino acid tagging, this is the substrate analog probing, where we feed the sample that came up under respective temperature pressure, this methionine analog. So it's a synthetic methionine that's going to be incorporated if the organisms are actually translationally active. So methionine is incorporated in protein synthesis. So these red bugs just indicate the natural community that's there. And then this green guy, let's say that he took up the methionine. We then are able to add chemical fluorophore through a sodium copper azide click chemistry reaction, where then an organism that incorporated the methionine will be fluorescent. You then can take these samples and subject them to FACS, where we sort out fluorescently labeled populations that are indicative of the organisms that are translationally active, and then compare that to the organisms that weren't. There's a couple of papers out on this right now, but they've all used 16S sequencing. So this paper that's in review right now is the first to apply it to actual metagenomic sequencing. So we took the cells that were sorted as translationally active, and we sequenced the metagenomes. Now these metagenomes, the results are a little bit better than single cell, than SAGs. And I think I heard Javier talking to you guys about SAGs before. But they're not great because we could only sort a certain number of cells. So on average, we sorted 300 to 5,000 cells and had many replicates, but this isn't enough biomass for metagenomics. So we had to subject it to an amplification procedure. So MDA, multiple displacement amplification. So what this takes away is our ability to say anything quantitative. All we could talk about was presence or absence. Are the genes there? Are they not? Who is there? Who is not? Which is why it was so important that we had those corresponding shotgun metagenomes, because then we can start to ask questions about which organisms are more dominant, and we can compare the organisms that popped up and those genetic functions in our Bongcat samples to the shotgun metagenomes. So after all of that effort, these are the, few, there was 12 Boncat mags that were above our threshold of 95% complete. And from the other ones, we just kind of neglected those for right now. We were just looking at the high completeness, low contamination. And what we did here was we phylogenetically identified those individual mags, but we also made our own gene databases, because for hydrocarbon degradation, as you know, Rachel knows with NIF-H, you have to sometimes scour the literature, make your own databases for the questions you want to ask. So had ideas about the genes that were maybe important in hydrocarbon degradation, but no databases. So the categories of genes that we were interested in are biosurfactant genes. Biosurfactants are really exciting because they're just starting to be recognized in the environment. They're important across all environments. They change the polarity or the ability of a microbe to interact with organic matter. You can think of it kind of as a detergent, where it can go in and it changes the chemical composition of something that organisms previously wouldn't be able to utilize. So it's really important for coal and bottom of the ocean. For carbon burial, biosurfactants are probably really important. They're really important in humics and streams. We then looked at some anaerobic hydrocarbon degradation genes, aerobic hydrocarbon degradation genes, and then of course our sulfate reduction. We wanted to know, were we getting presence of sulfate reduction, and then methanogenesis. And so in this graph, we see all of our different Boncat mags, and then the colors indicate which genes were present or absent. And we had these specific genes that we were looking for over here. Our list was like 200 genes long, but these were the ones that we narrowed it down to as being the most important. So then we took this information of the genes that were present in our active or translationally active about. And there's a lot going on in this, and a lot of genes, but essentially what I want you to take away from it is each different gradation of gray is the different wells that we sampled in the field. And if there's a star above it, it indicates that those genes were also found in that translationally active fraction. So we knew then to focus in on the genes that were in the translationally active fraction as being relevant for hydrocarbon degradation under the conditions that we sampled. And the genes that really stood out, because not much is known about them in the literature, and they had high abundances, were these biosurfactant genes. So our conclusions, because I'm probably close on time, is that we determined that biosurfactants are likely really important players in biogasification in this initial hydrocarbon degradation step for low sulfate environments. Previously, everybody thought it was rhamnolipids, but what our data suggests, and we did have rhamnolipids present, and we wouldn't have known that they weren't active if we didn't couple our methods with this active metagenomic sequencing, is that it's actually these glycosin and surfactant molecules that we want to investigate further now as to how are they functioning in the environment, that are probably really key in that rate limiting initial degradation step. And something else that popped out that I didn't mention here is that we talked about anaerobic, so this anhydeg, and aromadeg. So what this is short for is genes that are thought to exclusively exist in anaerobes versus be present in aerobes. Methanogenesis has been thought of for a very long time to be a solely anaerobic process, very sensitive to even trace amounts of oxygen. But what we're showing from our data is there's the presence in active fraction of aerobic degradation genes and anaerobic degradation genes. So that is an outstanding question that we have, is is oxygen actually involved in some way in the methane sequence production? We don't know. That needs further investigation, but it was kind of a interesting finding. And so then ultimately, how does this information that we obtained about these biodegradation steps, how are we going to then apply that back? And how are we going to actually in the real world enhance methane production? Well, we finished a field scale injection where we actually went back to the field, injected heavy water, a yeast extract substrate down our wells into the environment, kind of test some of these hypotheses. And then we monitored this for, about a year and a half, and we saw an increase in methane production that was about 1.32 times greater. This doesn't sound huge, but it was enough methane produced to, we calculated in the paper, support our university for an additional 6 months. And so this was just one small well, but I wanted to give you kind of a snapshot of how we're taking this fundamental, microbial research and then kind of turning it into an applied aspect by working with engineers and really trying to solve real world problems. I just want to thank our entire coal bed methane and a lot of work over the last 5 years.